Guys, today let's start to understand gluconeogenesis. Now, this is a process by which we produce glucose from non-carb sources, right? Non-carb means we have not eaten anything. So if we have not eaten anything, it means we have practically no insulin. If we have no insulin, when the glucose is gone, what hormone is secreted? Glucagon is secreted because glucose is gone. So this whole gluconeogenesis is done under the influence of glucagon, right? Let's try to understand gluconeogenesis, right? Now the conceptual overview is that why it even exists, okay? It exists because we need glucose, okay? We need glucose for everything. We need glucose for RBCs, we need glucose for brain, right? And what will happen if you don't eat for, maybe 3 days? This sort of pathway is going to take place so that we get some glucose out of non-carb sources. That's why it exists. It's done under fasting. It means it's done under glucagon. Liver and kidney cortex, right, plays a big role here, okay? Because liver is one of the biggest carb sources in our body, not carb, glucose sources, and kidney cortex is one of the big sources as well. That's gluconeogenesis for you. Literally, gluconeo means new, genesis means forming, and gluco means glucose. Glucose new forming. Okay, that's the logic. Now, why in kidney and why in liver? So kidney, it maintains the glucose level for your brain and RBCs, right? Sorry, liver, it maintains this. Kidney, it's like a backup for your fasting phases. And why not muscles? Muscles don't store this kind of things. Why? Because muscles don't have something called as glucose 6 phosphatase. So liver export glucose and muscle use this exported glucose. Okay, so step one is that glucose, right? Glucose turns into oxaloacetate. Sorry, pyruvate turns into oxaloacetate. Now, the enzyme here is pyruvate carboxylase, right? And the cofactor here is biotin, B7. And the activator here is acetyl-CoA, and the location where it happens is in mitochondria. And note, this whole process is a ATP, it uses ATP, so it's not very efficient, right? For example, in TCA cycle, we give 2 ATP and we get 4 ATP. So it's not this TCA cycle in itself is not a very efficient cycle. So the concept meaning here is that pyruvate is the end product of glycolysis, right? It's a 3-carbon molecule. Now, what happens is that this carboxylase, right? It adds carbon dioxide to make something called as oxaloacetate, OAA. Now, OAA is a 4-carbon, and this is a 3-carbon, right? Now, we have biotin, right? Biotin, it carries something called as CO2, right? And think of biotin as a CO2 carrier. That's it. Now, here, acetyl-CoA is an activator, right? Now, acetyl-CoA signals that, hey, we have enough fat. Now, it signals that, it says that we have enough fat burning, time to make some glucose. That's the concept here. Acetyl-CoA signals this. Now, why this makes sense is because when fat is being oxidized, when fat is being burned, acetyl-CoA builds up. Now, once acetyl-CoA builds up, it activates pyruvate carboxylase, this one, because fat is getting burnt, right? Fat is getting oxidized. Now, once this carboxylase is, activated, it directs pyruvate towards the glucose formation instead of TCA, because pyruvate can go to TCA cycle, but hey, acetyl-CoA levels builds up. Once acetyl-CoA level builds up, the carboxylase says, hey, don't go to TCA, come here, make oxaloacetate. So that's the concept here. Now, once oxaloacetate is formed with the help of phosphophenol carboxylase, right? Now, here the enzyme is phosphophenol pyruvate carboxylase. Now, once with the help of this enzyme, oxalate turns into phosphophenol pyruvate, okay? Now, here the enzyme is pep carboxykinase, right? And the cofactor is GTP, okay? The location here is cytosol. Now, this pathway, this conversion is happening in cytosol, okay? Okay, cytosol. Why? Because oxaloacetate must leave mitochondria as malate or as aspartate. That's the why this reaction is happening in cytosol. Now the concept behind it here is that carboxykinase, right? This phosphoenol pyruvate carboxylase. This whole enzyme. It actually adds a phosphate groups. It adds a phosphate group to oxaloacetate. From where does it gets the phosphate group? It gets the phosphate group from GTP. Okay. Now this GTP turns into GDP later. So what happens here is that GTP is used instead of ATP because GTP is the nucleotide energy for anabolic reaction, right? And this GTP is highly present into cytosol. So conversion of OAA oxalic acetate turns into phosphophenol pyruvate. This conversion is like releasing a spring that was compressed, and it's done. This compression was done using CO2, okay? Because carboxylase puts one carbon. This is a 3-carbon pyruvate, and oxaloacetate is a 4-carbon. So this 4-carbon adds one phosphate group with the help of GTP. Now we get phosphophenol pyruvate, right? This whole shit makes sense because adding carbon dioxide was the step 1, right? Now that's how the energy is stored, right? Now this whole step essentially launches carbon back into cytosol to climb uphill towards the glucose. So that's why it's happening. I hope I make sense. Step 3 is this phosphophenol pyruvate, right, turns into fructose 1,6 bisphosphate, right? Now the enzyme here are multiple enzymes, right? It's basically a reverse of glycolysis, right? Here we use enolase, imutase, aldolase, etc., right? It's not really important. One of the cool thing about is that phosphophenol pyruvate can turn into fructose 1,6 bisphosphate, and then fructose 1,6 bisphosphate can turn into phosphophenol pyruvate. Now this is a reversible thing. Okay, so that's the thing. The cool thing here is that the energy consumed in this process, right, is 2 ATP plus 2 GTP. That's the cool thing. Okay, now for every molecule of glucose we need this currency, 2 ATP, 2 GTP. Okay, so this is like energy is being used and it's being invested, right, to get this 6-carbon sugar, right? C6H12O. This is like glucose, right? For every glucose we need 2 ATP plus 2 GTP here. Okay, so it's like you give a little bit and you get more. That's it. The step 4 here is that fructose 1,6 bisphosphate turns into fructose 6-phosphate with the help of fructose 1,6 bisphosphate, right? Now this is the rate limiting step, and it can be positively influenced by citrate and negatively influenced by AMP or fructose 2,6 bisphosphate. Okay, now this is a rate limiting step. This is one of the most important steps. What here is happening is that this activation is done by citrate, right? So presence of citrate means the TCA cycle is full already, right? Because citrate is one of the production, right? So TCA cycle is already full. It means it's time to make some glucose. Okay. Another thing is that AMP, right here, AMP is a low energy, signal. It's mono, do you know ATP, right? ATP, ADB, right? And AMP. What is this? This is triphosphate. This is diphosphate, means 2 phosphate. And this is monophosphate. So which is the higher level? Which is the higher energy level? Of course, triphosphate, because it has 3 phosphate, right? So the energy is transferred in phosphate ways, right? If you give and you take phosphate and the energy is transferred. So if we have AMP, it means it's a low energy state, right? And what here is happening is that this fructose 2,6 bisphosphate is also an insulin signal, right? Now, I already told you that this whole shit is a fasting shit, right? This is happening when you're fasted. Now, if you have already an insulin signal, right, fructose 2,6-bisphosphate, right, how can this thing happen? This thing will not happen. This whole process will come to a stop, because we have something called as fructose 2,6-bisphosphate, which is a signal for insulin, right? So this happens under the influence of glucagon when glucose is gone, right? Because we are just changing non-carb into glucose. That's the concept. So this whole thing, fructose 1,6-bisphosphate turns into fructose 6-phosphate. This whole shit happens in cytosol. And this is fructose 1,6-bisphosphate is the rate limiting step of this whole process, right? This whole process. Okay, so this was this. Let's try to understand fructose 6-phosphate turns into glucose 6-phosphate, right? Now, the physical meaning here is that citrate means we need some, energy. We are dying of hunger. We need some energy. This is like, oh, we already have energy. It's like, right? This is negative. This is positive. That's why citrate positively influences fructose 1,6-phosphate. Okay. So step 5 is fructose 6-phosphate turns into glucose 6-phosphate. Now, a cool thing about fructose is this. Why do we need fructose, right? We know that glucose turns into fructose, which later goes into TCA cycle, and then which later gives us energy, right? That's the concept. Now, why can't we just right away use glucose? Because the structure of glucose is in such a way that it cannot be divided into, it cannot be efficiently divided into, parts that can be used for TCA cycle. But on the other hand, fructose can be efficiently divided into little parts, right? And that's how it can be used in TCA cycle. So that's the concept, because fructose spending fructose is more efficient for our body than spending glucose. Okay. So that's the concept between glucose versus fructose. So this whole thing is reversible, right? It's a standard pathway, right? A lot of glycolytic enzymes are used here. This is just a reshaping molecule, right? And it's very simple, right? So it's a reversible thing. It's not a huge thing. Now here, glucose 6-phosphate turns into glucose by using glucose 6-phosphate, right? Now, location here, this whole shit takes place in endoplasmic reticulum of liver, kidney, and this shit is absent in muscles because this enzyme, glucose 6-phosphate, is not present in muscles, right? Imagine if muscles are producing energy, then which part will use the energy, right? If muscles themselves produce energy, then which part is gonna use it? Because muscles should use it, right? So function is to remove this extra phosphate from glucose 6-phosphate and just get glucose, all right? So we use something called this phosphate, okay? Now it removes this extra phosphate, it frees the glucose, right? The concept here is that this phosphate group on the glucose 6-phosphate, it traps it inside the cell, right? It cannot cross it. Now this G6-phosphate, now the concept here is that if you have phosphate attached to you, you cannot go freely in the bloodstream. So you need to be free. You need to be free so that you can bind to other proteins, right? If you already have phosphate, how can you bind to other things, right? So this glucose 6-phosphate removes this extra phosphate. So that's the concept, right? So that's the whole gluconeogenesis, simply, shortly. And the whole thing here is to keep a good blood sugar levels, right? Euglycemia, it means uniform. And body needs glucose as there is fasting, and it's an emergency pathway. It's like, oh, we are running out of glucose. What are we gonna do? We're gonna do some gluconeogenesis. That's what we're gonna do. So that's the concept of gluconeogenesis. All right.